Okay, we're going to continue our discussion of passive transport, how things get into and out of cells through that semi-permeable cell membrane. Remember, there's three types of passive transport. There's diffusion, there's osmosis, and now we're going to get our third type called facilitated diffusion. Remember, all these types of passive transport do not require the cell's energy, and they always move things from a high concentration to a lower concentration. Our last type of passive transport is called facilitated transport. Sometimes it's called facilitated diffusion. To facilitate means to help something. So this is diffusion with some help. So we're still talking about small molecules just like regular diffusion. For some reason they can't get through that lipid bilayer like how oxygen could or how water can through osmosis. So they go through these little channels created by proteins. These molecules still go from a high concentration to a low concentration, and this requires none of the cell's own energy. The proteins that are making these channels are those transmembrane proteins, and this is just like when you're going down the turnpike and you go through that Lehigh Tunnel. You're going through this channel. They cut through that mountain. That's exactly what facilitated transport is. You're still driving down a road, but you can't get through that mountain range. The only way you're going through that mountain range is to go through the Lehigh Tunnel, and that Lehigh Tunnel is just like that channel created by these transmembrane proteins. So if you think of passive transport as going through a big tunnel, that's what it's like, but it's just regular diffusion, but you're going through a special protein that makes a tunnel through the cell membrane. So make sure you write down all of these details. And that's our last type of facilitated diffusion. Our last type of passive transport is facilitated diffusion. Now, the lab that you were doing last week, I could always ask you this question: where does the energy come from to move molecules from a high concentration to low? Well, remember, passive transport, the energy does not come from the cell. And that lab that you did, hopefully you're seeing that in the air, diffusion happens faster than it did in the water, and in the water it happens faster than it did in the gel. So you got to think about where is the energy coming from to move your food coloring around, or to move that air freshener around, or to move things back and forth in a cell. So that's what we're talking about with this stuff, and we'll talk about this again more this week. Now we're going to move on to our next type of transport, which is active transport. So we're still in section 7.3, and these are going to obviously be a little different than passive transport. So let's talk about this just a little bit. First off, you have to know how certain molecules get moved, because the cell would obviously rather move things passively because it doesn't require any energy. But sometimes things have to be moved through with active transport. So, molecules that are small and uncharged, they'll get through the cell membrane passively. And I gave you some examples here. Oxygen is small and uncharged. In your lungs, passive transport moves oxygen from your lungs into your blood. Water doing osmosis requires none of the cell's own energy, and that's passive transport. Carbon dioxide, amino acids, these are all things that the cell can move passively. Molecules that are big, still microscopic, but compared to an H2O, they're bigger, like a protein or something like that. And molecules with a strong charge, like an ion, like a sodium or a potassium, they have to be moved with active transport. Now this picture is perfect. Here you see your cell membrane, and you can see the phospholipids here. You see the other part of the bilayer here. You see some transmembrane proteins. Well, something like H2O, it can do osmosis, which is passive transport, and it can sneak right in between these phospholipids. So that's passive transport. Here's a non-charged molecule, relatively small. That can move through passively as well, right through those phospholipids. This is something big, where macro means big. Big molecule. This can't sneak through. The cell's going to have to put energy in to get that in or out, and that's going to be active transport. And up here, charged molecules and ions, strong negative charge, strong positive charge, they can't just sneak through the phospholipids. They're going to have to be moved actively as well. Here are some general characteristics of active transport. The cell's going to have to put its own energy in. The cell's energy is called ATP. Sometimes you're going to take molecules from where they're in a low concentration and move them to where they're already in a high concentration. That's the opposite of diffusion. That would be like taking all the air freshener molecules and putting them back in the can. You're taking them from a low concentration in the room and you're putting them back into the can when they're in a high concentration. It's almost like collecting things and putting them in one place. Sometimes the cell needs to do that, and that would be active transport. And like we just said, moving very large things into or out of the cell, that's going to take the cell's energy. That's going to be another type of active transport. So this is much different than passive, and we're going to start noting this. Okay, we have 3 types of active transport. One type is called molecular transport. Sometimes these are called pumps. These are going to be transmembrane proteins in the cell membrane. They're going to act as energy-requiring pumps to move small charged molecules across the cell membrane from a low concentration to a high concentration. So this is the exact opposite of diffusion. You're not going from a high to a low. You're going from one side of the cell membrane, there's not a lot of ions, and you're going to push them by using these pumps and using ATP up to where they are already in a high concentration. Our muscles do this, and it's called the sodium-potassium pump. Now you have to jot all that down, but let me play a video here for you just to show you this molecular transport, pumping ions from one side of the membrane to the other.
The sodium-potassium pump is an active transport mechanism that is driven by the breakdown of ATP and works through a series of conformational changes in a transmembrane protein. 3 sodium ions bind to the cytoplasmic side of the protein, causing the protein to change its conformation.
Sodium is a strong positive charge. It cannot get through the membrane by itself. Down here, this one called K+, that's potassium. The plus means it's also a very high charge. It's got a positive charge on it. That can't get through on its own either.
In its new conformation, the molecule becomes phosphorylated at the expense of a molecule of ATP. The phosphorylation induces a second conformational change that translocates the 3 sodium ions across the membrane. In this new conformation, the protein has a low affinity for sodium ions, and the 3 bound sodium ions dissociate from the protein and diffuse into the extracellular fluid.
You see what happened? We put those positive sodiums from this bottom side inside the cell, and we pumped them using ATP, the cell's energy, out to outside the cell. Your cells are doing this all the time.
The new conformation has a high affinity for potassium ions, 2 of which bind to the extracellular side of the protein. The bound phosphate now dissociates, and the protein reverts to its original conformation, exposing the 2 potassium ions to the cytoplasm on the inside of the cell. This conformation has a low affinity for potassium ions, so the 2 bound potassium ions dissociate from the protein and diffuse into the interior of the cell.
Okay, so those positive triangles, they had K+, those potassiums, they got pumped from this blue part outside the cell through this protein, used energy, and got pumped inside the cell. So that's what we're talking about. Sometimes a cell needs to do that. It needs to move things that won't normally be moved, and it does that by using active transport, these molecular pumps. Okay, another type of active transport is called endocytosis. Endocytosis is where the cell actually bends itself around something that it wants to be. And look down here at this picture here. It's actually going to push its cell membrane out, kind of like arms reaching out to give a hug to this thing. And then it completely encapsulates it and brings it in here as a vacuole. Remember, vacuoles are for storage. And that's how it can bring solid things in, like food particles or foreign cells or just something big that it needs to bring in. It does endocytosis. So take a minute, write down that. There's actually two types of endocytosis. There is, sorry, this up here should say two types. There's phagocytosis, and that means it's endocytosis of a solid. That's what that picture just showed. So if it's a particle, a big protein, another cell, the cell actually works like a blob and moves around it, pinches it off, and brings it inside itself. Pinocytosis is the same exact thing, but it's of something that's more of a liquid or an oil that the cell needs. So it bends around the droplet of whatever because it can't come in through passive transport, pinches it off, and it's in a little vacuole. Maybe the cell can use a lysosome or something like that to break it down. So these are up here again. This should say two types. Phagocytosis is the endocytosis of a solid. Down here, pinocytosis is the endocytosis of a liquid. Let me show you a video of this too. This is going to go quick. But this blob up here is a white blood cell. They're going to keep calling it a neutrophil. And it's going to kind of go around and eat some of these dark staining bacteria here.
The neutrophil becomes polarized and starts chasing the bacteria. The bacteria, bounced around by thermal energy, move in a random path, seeming to avoid their predator. Eventually, the neutrophil catches up with the bacteria and engulfs them by phagocytosis.
Alright, they sped that up quite a bit because it would take forever just to watch it. But here's the little bacteria, and you saw that this big blob-like cell just kind of moved itself around it, did phagocytosis, and is now completely surrounding that little bacteria. That's what our white blood cells do when we get an infection. They find the bacteria, blob around them, and then take them into their lysosome to destroy them.
Them by phagocytosis.
Alright, so there's an example of phagocytosis. Alright, our third type of active transport is called exocytosis. This almost has the word exit in it. This is the exact opposite of endocytosis. This is when something inside the cell has to get out. Maybe some proteins or some hormones or something like that that the cell has just made. So this is when a vesicle maybe comes off that Golgi apparatus, fuses with the cell membrane, opens up, and then can get outside the cell. So your pituitary gland is making growth hormone. When your muscles and bones need to grow, it will make the growth hormone, release it through exocytosis, and then it can go to your muscles and bones and other parts of your body and make you grow. So exocytosis is active transport where the cell's going to change shape to let something come out of it to go into its surroundings. Okay, so jot all that down. Here's a good picture of exocytosis. Here's your Golgi apparatus, vesicle, new made proteins in here. They come, fuse with the cell membrane, and it kind of opens up like a little pocket opening up, and it lets the stuff outside the cell. Same exact thing down here. That's exocytosis, the exact opposite of endocytosis. Okay, so there you have it. That is the last type of passive transport called facilitated diffusion, using a transmembrane protein to move things into or out of the cell without using the cell's energy. And then we said for charged things or very large things, we have to move them actively through molecular pumping endocytosis or exocytosis.